Hi everyone, I'm Eric Smith and this is
Rhett Mile.
and we are going to get you ready for taking the exit exam, particularly the biochemistry section of it. This is just a general breakdown of what we're going to cover real quick. It's going to be pretty fast paced and quick because we have to get through it, but it can generally be broken into acid-base chemistry, problem solving, which we're going to just look over some sample equations, and then a really big broad topic cram session over macromolecules, metabolism, and genetics.
Alright, so first we're going to do acid-base chemistry. You gotta read the joke?
Oh, and there was room down here for a basic chemistry joke, but we weren't sure if the reaction would be too positive.
So you got your strong acids that everybody should be familiar with. You got hydrochloric, hydroiodic, hydrobromic.
Nitric acid,
Then you got your perchloric
and chloric acid, and sulfuric acid.
And then also for bases, it's pretty much anything with a hydroxide group will get taken off. So you got your pKa values, which are the main ones you're going to work with, which are the acid dissociation constants. Your
Yeah, isoelectric are your PI values, and that is when the species in the solution, so the weak acid or base that's in the solution has a net charge on it of 0.
And you got your polyproduct, which is pretty much it has or can have more than one dissociation value associated with it based off how many hydrogens are attached and if they can be taken off or not. Then you have your buffers, which is everybody should be familiar with buffers. And they resist pH change within a certain range based off what you may come out of that's made of a weak acid and a strong base, vice versa. So problem solving.
Yeah, so the way we're approaching problem solving is solving problems the easy way with guessing, at least that's what we think, and or our guessing is the easy way. We might be wrong, but that's why we're here. So let's go ahead and look at, we're going to look at 2 questions from the sample equations that are given as like the sample questions for the exit exam. Specifically, we notice a trend about most of the questions that are directly related to biochem involving pKa values and PIs, just kind of roughly knowing those. This first one we're going to go over doesn't really cover that. It's slightly related, but the easiest way to do it is just to graze over it. So this one is asking, you're given the fluorescence of a standard at a certain concentration, and then they tell you your unknown intensity fluorescence is a certain number, and they ask you to find the concentration for it. So here we have the standard is 1 microgram per milliliter, and its intensity is 73, and the unknown's intensity is 62, and they want to know the concentration in micrograms per milliliter. This is a simple proportion, so you have the 1 microgram per milliliter, you put it over its intensity, you make it equal to your unknown value, which would be x, over its intensity, which is 62, you multiply, you divide, you get the answer real quick, it's 0.85. A little bit more complicated question that's involved with biochem are the pKa ones. So this number 5 is pretty early on in the sample questions. What will be the approximate pH of a buffer prepared by mixing 100 milliliters of 0.1 molar sodium phosphate, and 100 milliliters of 0.1 molar sodium phosphate, but they're at different species types. So one is singly protonated with 2 sodium ions, and then the other one is doubly protonated with only 1 sodium ion. You're given a list of pKa values. These are important because they will help you figure out where to start. So the pKa1 value is the lowest value, and that means that the species has all of its available protons, which means it's nothing but phosphoric acid. So it's H3PO4. It can also transition into the doubly protonated one, and that's where they'll kind of balance out down there. pKa2 is the same case, except you'll have the doubly protonated and the singly protonated. And then pKa3 is when you only have the singly protonated or not protonated at all. This question is easily solved by understanding that at these certain pKa's, certain species are going to be more apparent than the others, more abundant. And since we just said that at pKa2, the singly and the doubly protonated one will exist in kind of like an equilibrium situation, the answer is easily picked out by that pKa. It's 6.8. That is the pH of the buffer if you were to make it with those two species involved. So the easiest thing to do for these kind of questions is to just find out what species are being involved, figure out what pKa they'll be at, which we'll also discuss on trying to figure that out easily in the next question we're going to look over, and you'll be able to find the answer really, really quickly. The next question that was rather pertinent was question number 32. Which of the following amino acids has its isoelectric point at the highest pH? So you're given a bunch of structures and a list of pKa values, and go get them. So the easiest way to go through this is look at the structures. Find out where a species can lose protons to make it neutral. You know that at pi points, the substance is going to have a net charge of 0. So starting with A, this is a positive nitrogen. Take off a proton, it becomes neutral. That means that after you remove one proton, it is neutral. So after, or at least around its pKa1 is where its pi will be. Looking at B, you have two positive nitrogens, so you can take off two protons. That means that at least around pKa2, after two removals, its pi will be in that area. Going through C, D, and E, each of them can only remove one proton before becoming neutral, which means that their pi's are closest to their pKa1s. Going back to the question, it's asking which one will have its isoelectric point at the highest pH. The inherent answer is B, because its pKa2 would end up being around its isoelectric point. You just solve the problem in under a minute if you can do that mentally and just know what to do when you see something like that.
Yeah, just at a quick glance, if you can just kind of count the positive charges, you'll know that those are easily going to be able to come off, and just go with the pK associated with that.
Bang. Those three alone will help you get through this exit exam, at least the biochem portion. Right, so now we're going to go into macromolecules. And I've seen bigger, says the scientist. And macro only fools you into thinking that they're huge. Go ahead. Okay. Yeah. So the first one we're going to cover is lipids. We don't really talk about lipids much. Just in general, we kind of just know what they are. But their monomer form are fatty acids, which are long chains of hydrocarbons connected by a glycerin backbone. They are usually hydrophobic and or amphipathic, which means that they'll both have like a hydrophobic end and a hydrophilic end, which is the example of cellular membranes being hydrophobic on the tails, so they congregate together, while the hydrophilic heads are positioned to the outside where the water content is higher. They can be saturated or unsaturated. Saturated is completely singly bonded hydrocarbon chains. Unsaturated has a double bond in it, which will make it less rigid at room temperatures, which is where you get oils and things of that nature. And they are mostly involved in biological systems for long-term storage of energy for metabolism, because they can get broken off and made into sugars. Next relatively important one, nucleic acids. Everyone should be pretty familiar with these, but their monomeric form are nucleotides, which involves having a 5-carbon deoxyribose sugar, or in terms of RNA, it'd be ribonucleic sugar, or ribose sugar, not a deoxyribose sugar. Then it has a phosphate as well as the nitrogenous base that makes it a nucleotide. Like I said, they'll come in deoxyribonucleic acids, which contribute to DNA, and then ribonucleic, which is RNA. They form phosphodiester bonds to create a backbone, which is what chains them along into being single or double-stranded, depending on what you're looking at. And there's 4 different bases attached to the ribose sugar backbone, which is adenine, thymine, cysteine, and guanine. Cytosine. Cysteine, yeah. Cystine's an amino acid. And those are represented over there by A, T, G, and C. And then if you replace that amine group at the top of the cysteine with, I think it's an OH group, it becomes uracil, or a double bonded O, uracil. All right, carbohydrates. This one is really important, at least in biochemistry, this one in proteins. So their monomeric form are monosaccharides. Everyone's pretty familiar with that. We all know they're called saccharides. And then there are 3 different variations: the monosaccharide, the disaccharide, which is just 2, and then the polysaccharide, which will contain many of them. They're hydrocarbon chains that orient themselves into rings, most often usually seen in biological systems as hexoses or riboses, because those are the most sterically stable. They are hydrolyzed in the body through metabolism for energy by the breaking of their bonds. And they have chiral and enantiomers that are important to metabolism, and they're used by enzymes. So here in the carbohydrate tree, just kind of breaks it down. You start with just a basic 3-carbon sugar, which is a polysaccharide, and you can add a carbon in and start creating this giant network of different sugars, just depending on the chirality of the new sugar added. So just an example, you have erythrose and fructose. When you add this 4th carbon in, if the OH group is to the right, if you're looking at it like we are, it becomes erythrose. If you add it to the left, it's fructose. And they have D and L conformations, or enantiomers, where the very first carbon that is introduced will either have the OH group on the right when looking at it, which would be D, or to the left, which would be L. And the bottom carbon here, or carbon 5, once you get to hexoses, those are the only ones that will stay the same depending on whether they're D or L. This is actually a question in the exit exam. They ask you at which carbon in the aldohexoses, which we have pictured here, would they be the same as the D-glucose picture they give you? And the answer will always be the 5th carbon here, because that will never change as long as they're all D conformations.
Do you want to jump to that one?
Yeah. They can look at it. What number is it though?
It's number 28.
Number 28. So if you want to go look at that real quick, it's there in the sample questions that's provided to us on D2L. Go ahead and go to the next one. So here are some semi-important polysaccharides, or at least common ones that will be probably put on there if they are included. So there's sucrose, which is a disaccharide of glucose and fructose. Note here, I didn't have a trio of it, but there are ketoses, which have a keto group at the top part of the chain and form into ribose sugars, so 5-carbon sugars instead of 6-carbon, whereas the aldoses have an aldehyde group at the end of the chain instead. So glucose and fructose join to make sucrose. Fructose is the ribose sugar or keto sugar, and then glucose is this aldose sugar. Galactose and glucose combine to form lactose, and maltose is glucose and glucose, but I think that's a beta bond. So it just has a different bond formation between glucose. All of these are pretty important simply because these can all get introduced into glycolysis or into a metabolic pathway in some form in the body. So they might show up on the exam. They weren't in the sample questions, but they might. So it's good to be familiar with those. Starch and cellulose are probably the most commonly talked about polysaccharides that are strictly saccharides, because there are other things such as chitin, but that has the introduction of amine groups and stuff. So the starch is the one we can digest. It's just multiple chains of glucose with an alpha 1,4 bond between the glucoses, and they're chained. And then cellulose is a beta 1,4 glycosidic linkage between glucoses that we can't digest. And if that shows you how specific enzymes are with how these are shaped and how they can function on these carbs, that is a really big example of it, because all that's changed is the type of bond that joins them together, and we can't digest one from the other. All right, now this is the big winner of biochemistry. It's proteins. So the monomers are amino acids. There are 20 different variations of those. Or the side chains for those that we'll be kind of glancing over. Proteins are coded by DNA, and we'll go over that in genetics. And they're studied in biochemistry most heavily because of the expression of a protein's impact on an organism, and generally have 4 levels of structure, which would be primary, secondary, tertiary, and quaternary, which we'll talk about once we see the amino acids. Do what?
We could probably draw like two lysines back to back real quick. You want to give an example of it? Or you can look at the book. It's actually better than this.
Oh, that's true. We could probably draw. We'll
Draw
save
like a
it for
chain,
the end. We'll draw a sample amino acid chain to show you kind of what they look like together. So here are all the different type of amino acids with their side chains. They can be generally broken down into nonpolar side chains, polar side chains, and then there's 5 electrically charged side chains, which are broken into acid and basic, or acidic and basic. There are only 2 acid ones, and then 3 basic ones. And all of these have certain effects on either the shape of the final protein, or if not just the shape, also how they will end up functioning and interacting with whatever substrates they may actually work on. This would be the primary structure for proteins. It's just the amino acids themselves. Going into secondary is when these start to get chained together and start to form what's called alpha helixes or beta pleated sheets. Alpha helixes can be oriented to turn right or left. Usually in human systems, or at least like almost 100% positive, most organisms that are alive today, they are oriented to the right. So you can take your hand, kind of make an L, and if you twist your hand with your palm going towards your face, that's kind of how the alpha helix will turn in terms of structure. And then for the beta pleated sheets, they can either run parallel or anti-parallel with each other. Parallel sheets, they simply run in the same direction. And they have really large loops that will allow them to do that. Whereas anti-parallel, one coded sequence will go up this way. There will be a small looping section, and the opposite will happen for the next part of the chain that's continued on.